Sorry, time ran out and I didn't realize I got cut off. All right, so anyway, again, here's our middle ear, and consists of these 3 tiny bones which transmit sound energy from the tympanic membrane to the membrane known as the oval window. All right, this just talks about we also have an ability to, of course, control loud noises by reflexes, really that should be probably managed through the inferior colliculi in the midbrain. All right, so the inner ear, this is the point where we needed to get this sound energy, because this is where we're going to have receptors or sensors for the energy. And these, again, are mechanoreceptors, so they're getting moved by the vibrations being transmitted through, or begun and then transmitted through the middle ear. So these are a bunch of interconnected tubes or tunnels and chambers in the temporal bone. So we have a bony and a membranous labyrinth, are the terms we're going to use. We have to cut into these to see this, all right? And we're going to transmit sound energy from air medium to a liquid medium, because sound travels much more effectively in a fluid. So the cochlea is going to be for hearing, the vestibule is for balance, but again, we're going to transfer this change through fluids now, as opposed to through air, as we had been. So in the membranous labyrinth, so inside these bones, we're going to see, and we'll see this in lab, these soft tissue structures, tubes, and this, so epithelium and connective tissue, and these tubes are filled with this substance we call endolymph, so endo inside, and then there'll be perilymph, right? So spaces between the bony and the membranous is filled with a fluid called perilymph, and again, we have a couple models to try to illustrate this, but these are outer chambers, and then there's going to be inner chambers, and all of this is fluid filled. And so once we begin these vibrations at the oval window, they're going to be transmitted into fluid and bounce off another membrane known as the round window to help drive the stimuli towards the sensors. All right, so here we are in the cochlea. So for hearing, we've cut away and we've magnified this to look at this. So we can clearly see the bony, cochlear wall, the bony chambers, or bony labyrinth. Okay, and we're trying to get, here's the sensors here in this place we call the organ of Corti. All right, and we're going to, the action potentials initiated here will travel through the cochlear nerve, and it will combine with the vestibular that's coming from the vestibule. It's somewhere over here. I should say about over here, right? And this is going to be our vestibular cochlear nerve, which is the cranial nerve 8, as they're saying down here. All right? So here, again, here's our bony labyrinth, and then we'll have this, membranous labyrinth here, and divided into different chambers. So membranous here, bony out here. Okay, so this is showing you the membrane. So this is still considered up here bony, part of the bony, and this is considered part of the bony, but this is the membranous, the inner tube, if you will. So 2 chambers within the bony, and then 1 chamber here in the membranous. Okay, there is a partial membrane in this membranous labyrinth called the tectorial membrane, and this is where you see these specialized hearing cells, which are also hair cells technically. And then we're going to name all these parts here. So they're giving you a couple names here: the scalea vestibuli and the scalea tympania. Right? And yeah, you're going to have to know those, but we'll see those. You'll know them within the lab as well. So turns out the cochlea as a whole is you're seeing this bony or osseous labyrinth here, and it's shaped like a snail's shell, and then the membranous is here inside in the middle of this. Right? There's a thin bony shelf called the spiral lamina, which is really right about here, that kind of creates, which is where we find part of the hearing organ, or organ of Corti. Right? The shelf divides it into an upper and lower chambers or compartments. The upper is called the scalea vestibuli and starts at the oval window and goes to an apex. So if we unwound all this, wound up, coiled up tube, we would get a tip, which is right about here. So it goes to the apex, The lower chamber starts at the apex and goes to the round window. So they're actually connected to each other through this apex. So they are continuous, but they're separated as we've made this cut here, and we're not at the apex. So the scale, we have to know those and name those. Scalea vestibuli, again, and scalea tympani. And the chambers, again, are filled with perilymph. So perilymph found in here. So these are still considered part of the bony. And they have a membrane lining, but this one is entirely membranous. So it is a membranous labyrinth here. This is also, you're going to find out, is called a scalea. They call it a cochlear duct here, and that is an acceptable term, but this is also known as the scalea media, if you like that term better. And this is where we'd find the endolymph. All right, so again, within the cochlea, the membranous labyrinth is the cochlear duct or the scalea media, and it's between the scalea vestibuli and scalea tympania, and is filled with endolymph. It ends at the apex. It's separated from the scalea vestibuli by a vestibular membrane, which is what we're saying here. We're calling this the vestibular membrane, right? And from the scalea tympania by the basilar membrane, which they show you here. And I always remember this one because it's the base where the organ of Corti sits on it. And of course, the vestibular membrane is named because it's associated with the scalea vestibuli. So that's the way I keep them straight. All right, this spiral organ, or organ of Corti, spiral organ because it's sitting on that spiral lamina, is the sensory area, right? So if we really magnify this, we see again, we simply call these hair cells. They have these little modified cilia, and you've seen cilia when we looked at the olfactory epithelium, and our taste cells or gustatory cells are also kind of modified, right? Hairs, if they call them that, but they're cilia that extend out. And these cilia are actually stuck, let's say tether, right? Stuck in the tectorial membrane. Okay, so this shelf, right, this parcel shelf here. So these again are sitting on the basilar membrane, and they have the hair cells, and this is really the sensory cell. This is what's gonna, right, connect to the cochlear nerve and eventually become the vestibular cochlear nerve. So the tectorial membrane, again, is attached to the spiral lamina, this bony shelf, and it doesn't move. But what's going to happen is fluid in these chambers is going to start creating waves. And so when the basilar membrane, which does move, starts to vibrate with sound, we're going to see these hair cells bend. And it's actually the bending of the hair cells, right? Remember, they're mechanoreceptors, that initiates an action potential that opens the ion channels, right? So hearing, the process of hearing, shown here says, different frequencies of vibration move different parts of the basilar membrane. But ultimately, it's the basilar membrane where that organ of Corti, or the spiral organ, sits, that has to be moved if we're going to hear. Okay, so this is looking again at the transmission, and you should be able to follow this because I'm sure I'll have a test question on this to say, okay, walk me through where sound goes in order to eventually get an excitation or action potential. All right. So sound is moved in, the tympanic membrane vibrates in response, and this vibration is amplified and enters into the inner ear. All right. When we're inside the inner ear, right, so here's our organ of Corti. It's a very simple diagram to kind of just show you this whole cochlea all kind of opened up, as they're looking at it as unwound, if you will. So scalea vestibuli, scalea tympanii, right, and then this membranous labyrinth in the middle, right? So vibrations against the oval window set, which are right here, start a wave moving, all right? And the waves move through the scalea vestibuli, the scalea tympanii, and this bends the basilar membrane, right? And causes the hair cells here to vibrate, which then, right, opens an ion channel. When they're bent, the ion channels open and close, the cell membrane depolarizes, right? So while hair cells are not axons themselves, they are excitable and have neurotransmitters, which then do excite, right, synapse to neurons, right? So why do sounds have different intensities, right? If this incoming sound is at a specific frequency, right, as we talked about this in hertz, Basilar membrane moves and moves the tectorial membrane in the same region. So as the frequency of sound changes, different hair cells are activated. So we can't detect anything below 20 or above 20,000, right? And that's, again, this is the frequency of the vibrations. And we're going to decode that in the brain, specifically in the temporal region of the cerebral cortex, which is our primary auditory cortex. We're going to be able to decode these vibrations that have been converted to action potentials. All right, balance, which is our equilibrium. This is done through the other parts of the inner ear, the vestibule and the semicircular canals. They use a similar, hair cells with stereocilia that you're looking at here in these diagrams. Similar mechanoreceptor, as we did for hearing. They sense the head's position, because they are in the head, right? Remember, the location matters. And the head movement and whether the bodies are moving, in relationship to the head. So the head position involves a vestibule. So where is your head relative to your body? And is sensed by these structures called the utricle and saccule, which you can't see on your models. You just see the vestibule. And then head movement is through the semicircular canals. So here we're looking at this idea of the utricle and the saccula. They have this structure called the maculae, and the macula has this membrane over it in which these hair cells are embedded. So similar situation as we did with the tectorial membrane. There are little tiny rocks, otoliths, right here in this membrane that then, as your head moves and gravity affects these otoliths, they move and pull or bend, right, these hair cells, and that starts an action potential through the vestibular nerve, right? If we're in the actual semicircular canals, we have a very similar situation. It turns out that in the vestibule, you still have bony labyrinth, the outer part, and of course the membranous, which is composed of these macula, this macula here. And we are going to again move the hair cells in response to gravity, and that tells us the position of our head according to which cells get excited. All right, so here membranous chambers in the vestibule is the utricle and the saccula or saccule, and these have this specialized epithelium that we talked about called the maculae, which is surrounded by endolymph, right? So again, we'd be in this fluid-filled chamber. The maculae has the hair cells embedded in it, so this is a collective, and these are embedded again in this gelatinous mass that has otoliths. So that's basically what we call stationary or static equilibrium. Where is your head in position to your body, right? Now the semicircular canals are really responding to movement of the head. I mean movement and the body moving. So rotational. Or we sometimes call it dynamic equilibrium, right? So it's an extension of the vestibule. So here's our vestibule collectively. We get these actual canals. It turns out that they are oriented according to the 3 dimensions of the body. So 90 degrees to each other. We have transverse, we have sagittal, and we have coronal. So this helps us because we're 3-dimensional creatures moving in 3-dimensional space. So it refines our ability to figure out where we are, our position. So here, we talk about the ampullae, these swollen areas here, as the semicircular canals attach to the vestibule. All right, still filled with perilymph. Inside each is a membranous canal, and here we talk about the cristae ampullaris. So that's a sensory, the hair cells and the sensory organ are collectively called this. And so here's our this idea of the structure. And the gelatinous mass is the cupula over this, right? And this is all contained, again, would be found in the ampulla. And the stereocilia, again, of hair cells are embedded in this cupula. So instead of a gelatinous membrane with otoliths, we just talk about the cupula or the cupula. And so this is displaced because this fluid moves it. So as you rotate your head, right, this endolymph starts moving and causes a change or a bending, specific bending of the hair cells, right? So that will on the next video, we'll talk about some of your general senses, and then finally with sight.